Hello my dear friends, welcome to Biopsy. Today we are going to discuss about the hallmarks of the cancer. So there are different 8 hallmarks of the cancer. First is a self-sufficiency in the growth signals. Means cell will grow by its on and on signals. They will not dependent on others for the growth signals. They will synthesize for own growth signals. Means oncogenes are the cancer protein. They are responsible for the cancer. So oncogenes, which are going to transcript the oncoproteins, and that oncoproteins, they are responsible for the self-sufficiency of the growth signals. Second hallmark, it's insensitivity to the growth inhibitory signals. मतलब कि जो सेल होगा रेजिस्टेंट हो जाएगा ग्रोथ इनहिबिटरी सिग्नल्स को, वो रिस्पॉन्स ही नहीं देगा. मतलब सेल्स ग्रो करता ही रहेगा. सेल्स आगे के आगे प्रोलिफरेशन होता रहेगा और दे विल लीड टू द कैंसर. थर्ड वन, अल्टर्ड सेलुलर मेटाबॉलिज्म. ऑब्वियसली अगर सेल ग्रो कर रहा है तो उसे एनर्जी चाहिए. एनर्जी कहां से मिलेगी? अब नॉर्मल सेल के पास there are two mechanisms for the energy. One is a mitochondrial oxidative pathway and second one is a glycolysis. Abhi, in this case of the cancer, there is special effect, Warburg effect. What is the meaning of Warburg effect? Normally, cell will synthesize their own ATP, own energy by mostly mitochondrial oxidative pathway. Aerobic cell कहाँ से energy synthesize करेगा? Mitochondrial oxidative pathway से. But mitochondrial oxidative pathway, which is a time consuming. But in case of cancer, सेल को एनर्जी ज्यादा चाहिए, वो भी कम टाइम में, तो वो एरोबिक ग्लाइकोलिसिस को यूज करेगा. मतलब कि ग्लाइकोलिसिस जो पाथवे है, वो उसमें से हमें अराउंड 2 ATPs मिलती है, नेट 2 ATPs. सो वो ग्लाइकोलिसिस पाथवे को यूज करेगा. उसे बोलते हैं वॉर्बर्ग इफेक्ट, aerobic glycolysis pathway. It's a Warburg effect. Fourth one, it's a evasion of the apoptosis. Means, what is the meaning of apoptosis? Apoptosis, which is a programmed cell death. मतलब कि अभी सेल डेथ ही नहीं हो रहा है क्योंकि इवेजन हो गया है. Fifth one is a limitless replicative potential. Means immortality. Cell will become immortal. डेथ ही नहीं होगा. Sixth one, सस्टेन्ड एंजियोजेनेसिस. ऑब्वियसली अगर सेल को ग्रोथ करना है तो उसे न्यूट्रिएंट्स चाहिए. न्यूट्रिएंट्स कहां से मिलेंगे? ब्लड वेसल्स. तो ब्लड वेसल्स के लिए एंजियोजेनेसिस चाहिए. सस्टेन्ड एंजियोजेनेसिस, व्हिच इज अ कैरेक्टरिस्टिक ऑफ द कैंसर. सेवेंथ, इट्स अ एबिलिटी टू इन्वेड एंड मेटास्टेसाइज. मतलब कि मेटास्टेसिस, नॉर्मल स्ट्रक्चर्स, उसके जो भी पेरिफेरल स्ट्रक्चर्स होंगे उसको इन्वेड करेगी ट्यूमर और वहां से मेटास्टेसिस होगा. और लास्ट वन, ability to evade the host immune response. Normally in our cells, in our body, there are T cells, there are immune regulatory cells. They will regulate the normal cellular proliferation. अगर कोई सेल ऐसा होगा जो लिमिटलेस प्रोलिफरेशन कर रहा है, तो हमारी बॉडी की जो इम्यून रिस्पॉन्स है, इम्यून सिस्टम क्या करेगी? उसे इन्वेड करेगी, उसे it will lead to the death of that cell, जिससे वो ट्यूमर बने ही नहीं. लेकिन in case of cancer, they have a ability to evade the host immune response. मतलब कि अभी होस्ट का जो इम्यून रिस्पॉन्स है वो काम नहीं कर रहा है, सेलुलर प्रोलिफरेशन हो रहा है और ट्यूमर बन गई. So what we have discussed different 8 hallmarks of the cancer. One is a self-sufficiency in the growth signals. खुद के ग्रोथ सिग्नल बना रहा है, सेल रेजिस्टेंट हो गया है इनहिबिटरी सिग्नल्स से. देन उसने एरोबिक ग्लाइकोलिसिस स्टार्ट कर दिया है, एपोप्टोसिस हो नहीं रहा है, सेल इमोर्टल हो गया है, अभी ब्लड वेसल्स ज्यादा के ज्यादा बनने लगी है, मेटास्टेसिस हो रहा है और होस्ट इम्यून रिस्पॉन्स काम नहीं कर रहा. These are the 8 hallmarks of the cancer. And today we are going to discuss first, self-sufficiency in the growth signals. Now we are going to discuss first about normal mechanism of the growth signaling pathway. How the cell will respond to the growth signal. Whenever this is a growth factor, this is a growth factor. Whenever this growth factor will arrive at the growth factor receptor, it is a receptor for the growth factor. Whenever it is arrived to the cell, it will bind to the growth factor receptor. And after binding, there will be some molecular changes, there will be some transformation changes, and it will lead to the activation of the some of the proteins, activation of some of the pathway, and because of these activation, these activated proteins will go into the nucleus. These activation of some pathway, it is known as a signal transduction pathway. Signal transduction pathway. After signal transduction pathway, molecules, proteins enter into the nucleus. After entering to the nucleus, these are the nuclear regulatory proteins. The nuclear regulatory proteins, they are responsible for nuclear regulatory proteins. They are responsible for normal transcription of the human cell DNA. They are responsible for the transcription of the human cell DNA. So they will regulate the transcription. And after that, these DNA, it will undergo to the translation, transcription, then translation, and it will form the proteins. And these proteins, they will further go to the, it will lead to the cell proliferation. So it is a normal pathway of the growth signal. How the growth factor arrive, how they will bind to the cell, how signal transduction pathway occur, and it will lead to the nuclear regulatory proteins, and ultimately there will be protein synthesis. So if there will be alteration in any of this level, if there will be alteration in any of this level, then it will lead to the carcinoma. Like if there will be more and more growth factor, then more growth factor will bind to the cell and it will lead to the more protein synthesis. So first, self-sufficiency in the growth signals, growth factor ज़्यादा है. या फिर if there is a mutation in the receptor, normally क्या होता है कि there are some inhibitory molecules, it will inhibit the binding of the growth factor to the growth factor receptor. But if there is a mutation in the growth factor receptor, then more and more growth factor will bind to the receptor and it will lead to the cancer. Third thing, if there will be amplification of the signal transduction pathway, if there is an amplification of signal transduction pathway, then it also lead to the cancer. If there is an alteration to the nuclear regulatory proteins, न्यूक्लियर रेगुलेटरी प्रोटीन्स अभी म्यूटेशन हो गया तो वो रेगुलेट नहीं कर पाएंगे नॉर्मल ट्रांसक्रिप्शन दो, सो देयर विल बी कैंसर. सो एंड लास्टली, नॉर्मली सेलुलर साइकिल होती है, सेल साइकिल, which is regulated by the cyclins, and if there is any alteration, any mutation in the cyclins, then it also lead to the cancer. So these are the different types by which cell will gain self-sufficiency of the growth signal. So first, it is a growth factor. If there is a overexpression of the growth factor, like if this is a PDGF alpha beta, platelet derived growth factor beta, if there is an overexpression of PDGF beta, then it will lead to the astrocytoma. These we have to mug up only. If there is a overexpression of the FGF3, fibroblast growth factor 3, then it will lead to the osteosarcoma, stomach cancer, bladder cancer, breast carcinoma, melanoma. If there is a overexpression of the transforming growth factor alpha, then it will also lead to the astrocytoma. If there is an overexpression of the hepatocellular growth factor, then also it will lead to the hepatocellular carcinoma. So first we have done growth factor. If there is any overexpression of the growth factor, then it will lead to the tumor formation. Second thing, if there will be any mutation in the growth factor receptor, then it will lead to the also cancer. It is a EGF receptor family, endothelial growth factor receptor family, and gene named as a ERBB1, ERBB2. This is a very important gene for the MCQ. ERBB1 means erythroblastic oncogene. Erythroblastic oncogene 1 and erythroblastic oncogene 2. Erythroblastic oncogene 2, which is also named as a HER, human epidermal growth factor. So if there is any mutation in the ERBB1, then it is also lead to the breast carcinoma. Very potential MCQ. Second thing, if there is any mutation in the PDGF receptor, then it will lead to the glioma or leukemia. Now, third thing, signal transduction pathway. If there is any alteration in the signal transduction pathway, then it will lead to the overexpression of the nuclear proliferation. So there are two main signal transduction pathway. One is a tyrosine kinase dependent pathway and one is a non-receptor tyrosine kinase dependent pathway. So first we are going to learn about the what is a tyrosine kinase mediated and what is a non-tyrosine kinase mediated. See, tyrosine kinase receptor, they are KRAS, HRAS, NRAS, GNQ, GNAQ. These are the tyrosine kinase mediated. And due to the overexpression of these signal transduction pathway, there are different tumors. One is a colon, lung, pancreatic tumor, because KRAS mutation, which is a very, it is a most frequent, most common mutation in normal cancers. In all of the tumors, KRAS mutation, which is a most common mutation of the signal transduction pathway. So first we are going to learn about the different types of the transduction pathway. So we have a tyrosine kinase pathway, and second thing, we have a non-tyrosine kinase pathway. So first we are going to discuss about the tyrosine kinase pathway. See, here it is a growth factor receptor. Whenever there will be growth factor will arrive, then it will lead to the activation of the RAS. This RAS is a protein which is a membrane bound protein. In the state of inactivated state, this RAS will bind to the GDP, but after activation, these RAS will bind to the GTP. Whenever GTP will bind to the RAS, it will lead to the activation of RAS protein, and due to the activation of RAS protein, it will lead to the activation of the RAF pathway, and due to the RAF activation, it will lead to the MEP kinase enzyme activation, and which is lead to the activation of the transcription. So very easy, whenever growth factor will arrive to the growth factor receptor, it will lead to the activation of RAS. These activation of RAS, it will lead to the activation of RAF and MEP kinase, and it will lead to the activation of transcription. Also, this after activation of RAS, it will lead to the activation of PI3K gene, and due to the PI3K gene, it will also lead to the cellular proliferation. Due to the activation of transcription, MYC, which is a nuclear regulatory protein, and D-cyclin, which is also a cell cycle receptor, cell cycle regulator proteins, and due to the overexpression of MYC and cyclin D, there will be increased protein synthesis. There will be increased protein synthesis. So it is mediated through the tyrosine kinase receptor. It is the receptor, growth factor receptor of these type of pathway. They are called as a tyrosine kinase mediated because they have an intrinsic ability of the tyrosine kinase enzyme. But in case of non-tyrosine kinase pathway, so it is an example of chronic myeloid leukemia in which there will be non-tyrosine kinase mediated overexpression. Normally, we have a ABL oncogene on the chromosome number 9, and BCR oncogene, which is on the chromosome number 22. If there will be translocation between these two genes, it will lead to the ABL gene and a BCR gene fusion. If there will be ABL and BCR fusion, then it will lead to the expression of the activation of growth factor signaling pathway, because this fusion gene, they will synthesize one protein, one protein known as a fusion protein, and these fusion protein, they have an intrinsic ability of the tyrosine kinase. Ability. So here, this is a tyrosine kinase enzyme itself, but here, due to the fusion, there will be synthesis of one protein, fusion protein, and these fusion protein, they have a tyrosine kinase activity. Okay, so there will be difference between the tyrosine kinase and non-tyrosine kinase mediated pathway. So there are different examples. Due to the KRAS mutation, there will be colon, lung and pancreatic tumors. And the case of non-receptor tyrosine kinase, it is a most common example is a chronic myeloid leukemia. Third thing, it is a nuclear regulatory proteins. As we have already discussed, MYC, NMYC, these are the nuclear regulatory proteins. And due to the amplification of these nuclear regulatory proteins, it will lead to the tumor suppression, Burkitt's lymphoma. It is a most important MCQ. Due to the mutation of MYC gene, MYC, which is a nuclear regulatory proteins, it will lead to the Burkitt's lymphoma. And lastly, cell cycle regulators. We have already discussed, normally cell cycle, which is regulated by cyclin and cyclin dependent kinase. There are different 4 stages of cell cycle: G1, S, G2, M. In the stage of G1, cell will synthesize their regulatory proteins for division. Then G1 will enter into the S phase. In S phase, there will be replication of DNA. From S phase, it will undergo G2 phase. In G2 phase, there will be also protein synthesis for the metaphase. And then G2 will enter into the M phase, which is a metaphase, which is a mitotic phase only. So from G1 to S, there is one checkpoint known as a checkpoint 1. There is one checkpoint 1, and from G2 to M, there is one checkpoint which is known as a checkpoint 2. These two checkpoints, they are responsible for the regulation of cell cycle pathway. So from G1 to checkpoint 1, cyclin D and cyclin dependent kinase 4 and 6, they are responsible for the generation of G1 to checkpoint 1. And from checkpoint 1 to S phase, cyclin E and cyclin dependent kinase 2, which are responsible. From S point to the G2 phase, cyclin A and cyclin dependent kinase 2 and 1, they are responsible. And from G2 to M phase, which is a very important, cyclin B and cyclin dependent kinase 1, they are responsible from the G2 to enter into the GM phase. A very important thing, the most important checkpoint is a checkpoint 1. Most important checkpoint, which is a checkpoint 1. From here only, it will decide cell will undergo cell cycle or not. If there is any alteration, any mutation in the genes, then cell will not undergo into the cell cycle proliferation. So checkpoint 1, which is a most important checkpoint. So if there is any alteration in the cyclin D, then it will lead to the mantle cell lymphoma. and if there is alteration in the cyclin dependent kinase 4, it will lead to the glioblastoma. So till now we have discussed first hallmark of cancer. It is a self-sufficiency of signal transduction pathway. So we have discussed about how the normal growth factor curve arrive. It will bind to the growth factor receptor and how it will lead to the cell transduction system. We have discussed about different types of mutation. In the next video, we will discuss about the RB gene and P53 gene, which are the tumor suppressor gene, which is also responsible for the cell cycle regulation. So thank you very much for watching the video. Please do like, share and subscribe. Thank you.